Humans vary a lot when it comes to ability. Some of us compose songs that bring others to tears, some run like the wind, and others dream up new technologies that change the world. But that's only the tip of the ability iceberg. A few who live among us have claimed they have extraordinary abilities. Join us as we discover whether these individuals truly have super senses or are just super frauds. Light comes in through the cornea and pupil and hits the lens, the convex transparent disc that focuses that light and projects it onto the retina, which makes up the inner layer of the back of the eyeball. Your retinas are loaded with millions of photoreceptors which do the crucial work of converting light energy into the electrical signals that your brain will receive. These receptor cells come in two types, rods and cones. Cones sit near the retina center and detect fine detail and color. They can be divided into red, green, and blue sensitive types. Rods, on the other hand, are more numerous and more light sensitive, but they can't pick up color. Instead, they only register a grayscale of black and white and hang around the edges of your retinas. As many as 100 different rods may connect to a single ganglion cells, but because they are all send their information to the ganglion at once, the brain can't tell which individual rods have been activated. That's why they're not very good at providing detailed images. All they can really do is give you information about objects' general shape or whether it's light or dark.
Seeing color in total darkness, as Elise Johns claims that she can, is actually impossible due to the anatomy of the eye. Cones which detect color only reach activation threshold in bright conditions. And as for the rods, you only have one type of cell, so there's only one type of signal that gets sent to the brain, light or no light. Having only one type of light detector doesn't leave any room for seeing color.
First, a stimulus activates some of your sensory receptors. In the case of stepping on a tack, it's a change from the probably a completely ignored feeling of bare skin on a smooth floor to a distinct feeling of discomfort. Your peripheral nervous system's mechano and nociceptors provided that base sensation or awareness that something had changed. Then it went to your central nervous system, first to the spinal cord that caused the immediate reflexive action of pulling up your foot, and then your brain eventually interpreted that awareness into the perception of pain and decided to pull the tack out. Pain itself is a pretty subjective feeling, but the fact is we all have the same pain threshold. That is, the point where a stimulus is intense enough to trigger actual potentials in those nociceptors is the same for everybody, but you and I might have different tolerances for discomfort. In general, most doctors think of pain as a perception of pain, whatever any given brain says pain is.
I'm here with Jeff Young, who claims that he can withstand any amount of pain. But withstanding pain without adverse consequences is impossible. The nociceptors can be divided into two groups, the A delta fibers and the C fibers. A delta fibers produce the knee-jerk reaction in response of immediate injury, such as pulling your hand away from a hot stove, while C fibers cause long-lasting throbbing pain, like the burn sensation you feel after your hand is burned. These nociceptors cause the sensation of pain to either be immediately felt with intensity or with continued enduring pain. However, the A delta fibers are what cause this supersense to be impossible, as the body will force itself to move away from the painful stimulus to decrease and prevent damage to oneself.
When you sniff molecules, most of these molecules are filtered out on the way up your nasal cavity as they get caught by your protective nose hairs. But if you make it all the way back to the nose and hit your olfactory epithelium, this is your olfactory system's main organ, a small yellowish patch of tissue on the roof of the nasal cavity. The olfactory epithelium contains millions of bowling pin-shaped olfactory sensory neurons surrounded by insulating columnar supporting cells. Once in the mucus, they're able to bind to receptors on your olfactory sensory neurons, which, assuming they hit their necessary threshold, fire action potentials up their long axons and through your ethmoid bone onto the olfactory bulb in the brain. But here's the wonder of specialization for you. Each olfactory neuron has receptors for just one kind of smell. So after each smell-specific neuron is triggered, the signal travels down its axon where it converges with other cells in a structure called a glomerulus. Inside the glomerulus, the olfactory axons meet up with the dendrites of another kind of nerve cell, called a mitral cell, which relays the signal to the brain. So for each mitral cell, there are any number of olfactory axons synapsing with it, each representing and identifying a single volatile chemical.
Alright, well I am here with Apex, who claims that he can smell better than a dog. I'm also here to prove that that is incorrect. Roughly 12 million specialized cells are located in the human's nasal cavity. That is the number of types of chemical compounds that would stimulate the human olfactory system, enabling a person to smell. All other chemical compounds without a specific receptor in this system will go through undetected by the person. In order to be able to detect a wider range of smells, new types of chemoreceptive neurons would need to be built, along with entirely new synapses that can carry the action potential to new sections of the brain dedicated to distinguishing and processing these new kinds of molecules. Building these new organs require an immense amount of genetic mutation, which is theoretically impossible.
The pinna is made up of elastic cartilage covered in skin, and its main function is to catch sound waves and pass them along deeper into the ear. Once a sound is caught, it's funneled down into the external acoustic meatus, or auditory canal, and toward your middle and inner ear. Sound waves traveling down the auditory canal eventually collide with the tympanic membrane, which you probably know as the eardrum. When the sound waves collide with the eardrum, they push it back and forth, making it vibrate so it can pass those vibrations along to the tiny bones in the middle ear. Now the middle ear, also called the tympanic cavity, is a relay station between the outer and inner ear. Its main job is to amplify those sound waves so that they're strong when they enter the inner ear. Gravity focuses the pressure of sound waves so that they're strong enough to move the fluid in the inner ear, and it does this using the auditory ossicles, a trio of the smallest bones in the human body, the malleus, incus, stapes, commonly known as the hammer, anvil, and stirrup. One end of the malleus connects to the inner eardrum and moves back and forth while the drum vibrates. The other end is attached to the incus, which is also connected to the stapes. Together they form a kind of chain that conducts eardrum vibrations over to another membrane, the superior oval window, where they set that fluid in the inner ear into motion. An important membrane in the ear is the basilar membrane, a stiff band of tissue that runs alongside the middle fluid-filled chamber. It's capable of reading every single sound within the range of human hearing and communicating it immediately to the nervous system, because right smack on top of it is another long fixture riddled with special sensory cells and nerve cells called the organ of Corti. So when your ossicle bones start sending pressure waves up the inner fluid, they cause certain sections of basilar membrane to vibrate back and forth. This membrane is covered in more than 20,000 fibers, and they get longer the further down the membrane you go. Kind of like a harp with many, many strings, the fibers near the base of the cochlea are short and stiff, while those at the end are longer and looser. And just like harp strings, the fibers resonate at different frequencies. More specifically, different parts of the membrane vibrate depending on the pitch of the sound coming through. So the part of the membrane with the short fibers vibrate in response to high frequency pressure, and the areas with the longer fibers resonate with lower frequency waves. This means that all the sounds that you hear and how you recognize them come down to precisely what little section of the membrane is vibrating at any given time. If it's vibrating near the base, then you're hearing a high frequency sound. If it's shaking at the end, it's a low noise.
I'm here with Ellen Pang, who claims that she can hear more sounds than a normal human can. However, this is impossible because the human ear is capable of hearing a minimum of 20 hertz or vibrations per second and a maximum of 20,000. Most people's range of hearing falls somewhere between this limited scale. However, it is physically impossible for humans to hear vibrations below 20 hertz or above 20,000 due to the anatomy of the ear. The cochlea, and specifically the cilia within the cochlea, cannot vibrate with such low stimulus, and therefore the neural impulse cannot be sent. With vibrations exceeding 20,000 hertz, the human ear is unable to transmit neural signals of such high frequencies because of the basilar membrane. Any vibrations above 20,000 hertz would not be able to cause the basilar membrane to vibrate because there would not be a place on the membrane that could detect and respond to such high frequencies. This goes for low frequencies as well. The portion of the basilar membrane farthest away from the stapes registers extremely low frequencies, and any frequency below 20 hertz is unable to activate this region.
When you take a bite, all the sensory information in there is quickly sorted by the 10,000 or so taste buds covering the tongue, mouth, and upper throat. Most taste buds are packed deep down between your fungiform papillae, those little projections that make your tongue kind of rough. The taste buds are tucked into tiny pockets hidden behind the stratified squamous epithelial cells on your tongue. Each bud has 50 to 100 taste receptor epithelial cells which register and respond to different molecules in your food. These epithelial receptor cells come in two major types: gustatory, or the kind that actually do the tasting, and basal, the stem cells that replace the gustatory cells. Every gustatory cell projects a thread-like protrusion of the cellular membrane called a gustatory hair, which runs down to a taste pore, a small hole in the stratified squamous epithelial covering the taste bud and the rest of the tongue. In order to taste a bite of food, those food chemicals or tastants must dissolve in saliva so they can diffuse through those taste pores and bind to receptors on those gustatory cells, and then trigger an action potential.
I'm here with Sydney, who claims that she can taste more than any other human. But that's just not true. All humans are limited to 5 taste sensations: sweet, sour, salty, bitter, and umami. Every human mouth averages 10,000 taste buds. Each taste bud is home to roughly 100 taste cells. Each taste cell responds to only one of the sensations. So what does this mean for extreme tasting? It means that the brain is only familiar to sensory impulses triggered by these senses, meaning any other type of signal sent from a super taste bud would not be understood by the gustatory cortex. It would be like trying to understand someone speaking a different language. Unfortunately, super tasting is impossible. Our taste buds will never be able to experience sensations other than the 5 depicted above. Although this may seem like a letdown, there are always new chemical combinations to try that will stimulate your existing taste buds. The possibilities are limitless.
So, do super senses exist? Based on the anatomical and physiological limitations of each of the senses, it should be no surprise that these super senses are in fact super frauds.